Okay, I wanted to start today with a little public service announcement. So it's that time of the year to start thinking about getting a flu shot. I got my flu shot at the same time I got my Omicron booster shot last weekend. And so I would encourage you to think about that, doing that if you haven't done so. They were both free. I was a little sore, I was a little tired, but they are worried. Now, they've said this the last 2 years, get your flu shot, because they're worried it was going to be, you know, really bad to have bad flu and bad COVID at the same time. You know, they're saying that again, I think this year, because we're not all masked up all the time, that it really could be a bad flu year. So at least think about, you get your flu shot and you still get the flu, you'll probably get the flu a lot more mildly. And of course, it protects you from even getting it, so certain strains. But we'll talk more about vaccinations later on in the semester, but I just wanted to plant that seed. Before we pick up from where we left off last time, everybody has taken the exam now. So let's take a few minutes to talk about the exam. This is one of the reasons that I make the first exam worth a little bit less. Before I curved it, the mean was a 70. And then the curve that I applied, I took the number that you missed and multiplied by 0.9 instead of 1. And then I added a point to everybody. So with that rather creative curve, that brought the mean up to like 74.5, so almost a 75. So that, you know, like I said, so it's worth 80 points. Your other exams will be worth 100. The finals weighted a little bit more. So if you did poorly on this first exam, it's not the end of the world, but you want to think about what you need to change for the next exam. So maybe you need to find some people to study with. Maybe you're just missing things. Maybe you need to study a different way. Instead of just, you know, reading over your notes, maybe you need to find a little bit more active way to start testing yourself and, you know, reviewing things. You know, for a good first place to start is to come in and look over your exam. I'm going to talk about the 5 most commonly missed questions. Feel free to come in, ideally during office hours, but maybe we can find another time that you can come in, look over your exam, see what you missed, and we can see if there's anything in particular that maybe is a pattern that we could address. So keep at it again, and hopefully we can improve if you weren't happy. So these were the 5 questions that less than 50% got correct. Which of the following are never found in both prokaryotic and eukaryotic cells? So this is just kind of, do you understand the difference between a prokaryotic and a eukaryotic cell? And we talked about that a number of times. Both prokaryotes and eukaryotes have ribosomes. They both have cell membranes. Both those things are absolutely necessary to a cell structure. They have to have an intact membrane. They have to have ribosomes to make proteins. Prokaryotes or eukaryotes may or may not have cell walls. They may or may not have glycocalases. Okay, lysosomes are only in eukaryotes. They're a membrane-bound organelle. And I think, you know, what we're seeing is some little basic skills that maybe weren't hammered in as well. Maybe we can blame the pandemic. I don't know, but that's kind of something that I would have considered a review even before you got to this class. This was one, a couple of the people that only missed a question or two, this was one that those 2 people missed. And I didn't, sometimes I read a question and I don't realize that it's difficult. So what are cilia similar in structure to? And so first of all, if you knew that cilia were only found in eukaryotic cells, okay, then you can cross out the gram-negative here or the prokaryote. And the other thing that if you knew that fimbriae were only in prokaryotic cells, then you should be able to narrow that down that way. And the additional thing that would have helped you if you understood that cilia are made of microtubules, just like eukaryotic flagella. Okay, so three different pieces of information that hopefully led you to that answer there. Again, it's like I think it's like 33% of you got that one right. Maybe only like a third of you got that one right. This one, I think a lot of you, we talked about several times how agar is a solidifying agent. I kind of asked this one in a little different way that maybe made you think a little bit. If a mutation allows a plated bacteria, so a bacteria growing on a solid medium, to gain the ability to metabolize agar, what will happen? Well, just because, so if you knew that agar was a solidifying agent, just because it liquefies, that's not going to kill the bacteria. Bacteria would still grow, but again, the media is changing. The best answer here is that the culture medium would liquefy. Remember, agar is not a nutritional source, so it's not going to make them grow any better because they're not getting any nutrients out of that. So best answer there was B. Certain bacteria release endotoxin when the cell dies. What's the source of endotoxin? Endotoxin is also, another name for endotoxin is lipid A, and that is found in the lipopolysaccharide, which is found in the gram-negative cell wall. So you did kind of have to go through a number of hoops to get there, but again, hopefully you could kind of, well, I know it's not this, and I know it's not that. Okay. And then this was the toughest question. Only about a quarter of you got this question right. Which of the following would be considered the most desirable resolution? So the best resolution is the smallest distance. Okay, so we're looking for which of these is the smallest unit of measure and the smallest distance. So if you weren't paying any attention to the units, then you might have saw the 0.02 and you might have chosen that one. But you got to pay attention to the units as well. Obviously, meter is really big, centimeters is really big. Nanometers, 2 nanometers is the smallest distance here. If this was 0.002 micrometers, that is equal to 2 nanometers. So that was the closest answer. But nano billionth of a meter, micro millionth. All right, so, all right, understandably, that one was a little bit more difficult. All right, so again, if you have more questions about anything, what you got wrong, a question you thought maybe wasn't fair or whatever, don't be afraid to come see me, ideally before the next exam, okay, about that exam. And I'm still kind of working, I've pretty much got the homework graded, but I still, there's some things I haven't figured out about how the website figures out that homework grade. So I'm still kind of working through that a little bit. So hopefully, if you can, are you able to see how you did on the homework? No, okay, yeah. So I want you to know like how you're doing on that. So hopefully I'll get that ironed out in the next few days. So where we left off last time, we were into chapter 7 about growth. And we started out talking about kind of how populations in general grow. Bacteria grow exponentially, meaning with every generation that they're going to have double the number of cells than what they did the generation before. So we talked about how we can kind of estimate that mathematically. And as we finished here, I gave you a sample question and kind of talked through how you could figure that out using this equation back here. Really, if I gave you a question like this on an exam, it would be a question that you did not need a calculator to use, because I don't like to have calculators out on the bench. So it would be something that might be best to just think through this way. If you're just thinking, okay, the next generation, I'm going to have twice the number, next generation, doubling it. So it should be pretty straightforward, but again, there's a couple of questions on your homework to practice that. So moving forward, we want to think about, okay, what are some ways that we count bacteria? And then we're going to think about ways that influence the generation time, or how quickly bacteria grow. Things like what temperature is their environment, what's the pH of the environment, things like that. Okay, and there's lots of terminology to go along with that. But if we think about measuring bacterial growth, if you're in lab, we already have done a lab that counts cells a couple weeks ago now. So hopefully at least a little bit of this might look familiar. There are two methods that require incubation in order to estimate microbial numbers. So the one that we did in lab was this viable plate count. We'll also talk about membrane filtration. So I'm going to come back to the slide in a minute and just write a few things about that. But what are those two methods, again, that require incubation? This is a schematic of the viable plate count. So what I don't like about this drawing is that it doesn't really, it's not clear, well, wait, what are you trying to count? So say you have a test tube with some bacteria in here, okay? And if you wanted to estimate numbers there, you could take out an aliquot of that test tube and spread it around on an agar plate. And then you assume that each colony that grows up started out as a single cell in that original culture. The problem with that is that there's going to be way too many cells in this original sample. You would probably just get kind of confluent growth on that plate. So we have to dilute that out so we get a countable range of colonies. So what we did in lab, we made a series of 1 to 10 dilutions and took out an aliquot. We did, I don't know what they're doing in this picture here with that loop. We used a glass hockey stick spread plate to spread that around. And then we counted the number of colonies that grew on that plate. So here we would want to use this plate to extrapolate back to that original numbers in the sample. And again, if you're in lab, you learned how to do that. You have to take into account how much of that dilution that you put on that plate to begin with, and what this dilution was. Okay, so I'm not going to go through a calculation here. You don't need to know that for lecture, but you're going to want to review that if you're in lab for your exam this week. The other way that involves incubating cells is a membrane filtration. So you would use this method if instead of having a really concentrated solution of cells, if you had a very dilute. Okay, so if you had a dilute suspension of cells, maybe so dilute that if you plated out that original sample, it wouldn't give you a countable range. It would be too few. So when does this happen in nature? Not very often. Oftentimes maybe for water samples. I did this once with a sample of water from the Grand River, thinking I had to dilute it a bunch to get a countable range. No, I ended up, sorry, and I think I would have to concentrate it. I ended up having to dilute it a whole bunch because there's a lot of stuff growing in the Grand River. The only time you might need to do this probably is if you are testing your tap water. Because again, we treat our water that we drink, so there's not a lot growing in it. It's not sterile, but there's not a lot there. So the way that you would do this, you would take whatever a known amount of whatever water that you're testing, and you put a membrane filter right here. So it's essentially a piece of paper that has really small pores of a known size. When you pour your water, and usually have to kind of suck that water through with a vacuum to get the water to come through that membrane pore. And then you put that membrane pore on an agar plate where the nutrients can kind of soak right through that membrane. You incubate it, and then you count the number of colonies that grow up there. And from taking into account how much water you poured in there, you can extrapolate back to how much is in your original sample. So they're doing this kind of thing all the time at sewage treatment plants and making sure that they're actually cleaning the water like they should be. So with these two methods, they both require incubation. So the drawback with both of these is that it takes time. It takes longer than some of the other methods that I'm going to talk about, because you at least have an overnight incubation, assuming the cells you're trying to count grow up pretty fast. So you don't get your numbers back right away. But one advantage is that you are only counting the cells that are alive, which usually is the only ones that you really care about. Okay, only a colony, only cells that were alive in your original sample will divide lots and lots of times to give you a colony that's growing on the plate. And both of them are pretty inexpensive. You're not using any really fancy lab contraptions, things that you wouldn't find sort of in a normal lab. So they're pretty inexpensive. That's a drawback as well. I mean, an advantage as well. Some other methods. You could use something called, sometimes we call this a Petroff-Hauser chamber. It says that in the fine print there. I often refer to it as a hemocytometer because it's just a little bit easier to remember that term, hemocytometer. But this is basically a thick glass slide that you can use. You would put a cover slip on it, something like this, and then your suspension that has the cells in it that you're trying to count, you kind of put in these little, there's a little groove right there. It doesn't show up very well, but it wicks underneath the cover slip, and then this kind of on one side or the other, there's a grid etched in there that you can see when you look at your cells under the microscope. And then you just count, you zoom in on a certain number of squares, and you count the number of cells in those squares. So then, like, knowing how much you've diluted your sample, you can mathematically extrapolate back to how many cells are in your sample. Okay, so that's you get your answer back right away. You still have to have cells kind of be in a countable range for it to be statistically significant. This is easier to do when you have larger cells, like red blood cells or yeast cells. It's a little bit harder to do with bacterial cells. You can't tell if they're dead or alive unless you put a certain dye on there that only live cells will take up. And then your book calls this an indirect method using a spectrophotometer. Indirect because it doesn't give you actual numbers. If you remember, this is what the spectrophotometers look like that we still use in Bio 120, I believe. I taught that class a long time ago. But remember, you put your sample in there and then you shine a light through at a certain wavelength, and it can quantitate basically how colored your solution is, or in this case, we call this turbidity, right? When we got lots of cells in there, then they're going to look a little bit cloudy. Okay, the more turbid they are, the higher the absorbance is going to be. Okay, so we can kind of at least very easily quantitate whether we've got a lot of cells in there or a little. Of course, live and dead cells are going to scatter light the same way, so we can't tell if they're dead or alive. Again, we don't get actual numbers. We don't get. Okay, so we get an absorbance reading. If we wanted to get actual numbers, then we would have to make up some kind of a standard curve using another method to count cells. So basically you take out the absorbance, oh, it's 0.3 I'm going to take out an aliquot and count that by a hemacytometer. So then I know, oh, 0.3 corresponds to whatever, 8 million cells or whatever. Make a standard curve, and then the next time, oh, my absorbance is 0.6, then I know, okay, that corresponds to whatever number of cells. So, and this is just the textbooks kind of a little more involved or a little different way to explain that. How in the spectrophotometer, you're shining white light. There's usually a certain wavelength we use that's around, I can't remember, it's kind of in that greenish yellow wavelength. It's like 320 nanometers or something. That's kind of the typical wavelength we use to get optical density, we call it. And then again, absorbance is going to be higher for a cloudy sample. It's a nice method to see if your cells are actively growing over time. We're getting a little bit more expensive range now. A Coulter counter. This is an electronic cell counter. A little bit more expensive here. We have one in the upper microbiology lab. It's a little box. It's not very big. The principle this works on that bacteria will disrupt an electric field. So basically, when your cells are in a solution and you kind of pump them through, you got to regulate the flow rate, so it's kind of, you know, not too fast or not too slow, and it will just, each cell will disrupt an electric field, and then you get your counts right away. Big drawback here is bacteria cells tend to clump together. So that can be a problem, and it can cause your counts not to be as accurate. So cell clumping can be a problem. And like the spectrophotometer, you know, dead and alive cells are going to both set off that disrupt the electric field. So both dead and alive are counted. And again, we usually only care about the live ones. Getting to like the most expensive, the most accurate, the top of the line, you really need super accurate results, is something called a flow cytometer. Now we're getting into a model with sort of like all the bells and whistles is going to be hundreds of thousands of dollars. They use a flow cytometer uses a light sensitive detector. You have to stain the cells that you're trying to count with a fluorescent dye, or maybe tag that fluorescent dye to some antibody that's going to be specific for the cells that you're trying to count. The flow cytometer not only will count those cells accurately, but it can also separate out the different populations. So in this example, this was just an example that I got from searching the internet, what some data looks like. But I think in these experiments, they were trying to quantitate the different kinds of cells in some seawater. So there's some different kinds of, I don't know, plankton or whatever here. So there's 3 different kinds, the green dots and the red dots and the blue dots all signify a different kind of phytoplankton or bacteria that they were trying to count. And again, you can separate them out. You can tell if they're dead or alive. So it's a, it's a very powerful tool. Where I went to do where I worked in a lab for a while, they had a PhD, a full-time PhD that was employed to help other investigators with their work if they wanted to use the flow cytometer. So it's complicated. The results usually come back in some kind of histogram form like this. So this was an example from your textbook. If you're interested, looks like they were counting some T cells, ones or maybe ones that expressed a certain marker and ones that didn't. So, so that's kind of our cell counting from, you know, starting with viable plate count, which is maybe kind of the more primitive, yet still important, can still be powerful, all the way up to using a flow cytometer to count cells. All right, so let's move on now and think about some of the environmental factors that are going to influence how quickly cells are growing. And if we understand these environmental factors, then that helps us be able to grow up things in the lab, because we know what kind of temperature they need, or oxygen concentration, or what pH we need to grow them at. So it also helps us know how to kill cells if that's our goal to do that. All right, so the first thing that we always think of, the easiest thing to manipulate a little bit, is the temperature. All cells are going to have an ideal temperature that they're going to grow at, but they will usually grow over a fairly large range of temperatures, maybe 20 degrees, maybe 40 degrees even. And oftentimes, when they're growing at their optimal temperature, like that means they're growing the fastest. And usually when you just get a little bit higher than that optimal, then they'll start to not grow. Enzymes will denature, and that's not going to support life. So usually we see after that optimal temperature, we get kind of a dive in terms of whether they're surviving or not at all. Here's just an example of some E. coli that was plated out about the same, three medias that are the same. The only difference was that they were incubated at a different temperature. We know that E. coli grows really ideally, like a lot of other pathogens, other organisms that live in our body. They grow ideally at body temperature. So 37 degrees Celsius, that's body temperature. They grow really fast. But if you incubate E. coli a little bit cooler, maybe they grow not quite as quickly. If you incubate them just at room temperature, they grow a little bit. If you kept incubating them for like a week, then they would eventually catch up to, the growth that you saw at 37. But they're just going to grow a lot more slowly at room temperature. And of course, we like to assign some terminology describing the optimal temperatures that organisms grow. Organisms that are psychrophiles, those are our cold-loving organisms. So sometimes you see different ranges, like different numbers describing their range, usually maybe less than about room temperature. Is when they're going to grow best. So there's algae that grow on polar ice caps, for instance. They would be considered psychrophiles. Mesophiles, these are most of the organisms, pretty much all the organisms that we work with in lab. Usually we would say they would have a range between room temperature and body temperature. In terms of what they'll grow. Those are mesophiles, and then thermophiles, usually between 40 and 80 degrees Celsius. These are our organisms that are growing in hot springs, places that are hot all the time, not just like, on a hot sunny day or something, maybe in your hot water heater at home even. And then there are even organisms that can grow right near boiling or even at boiling. So again, thermal deep sea vents, hot springs, places like that. Seems like I was going to say something. Oh, and there are all these little kind of subcategories. Like there are organisms that they grow pretty well in the refrigerator. So that would be at like 4 degrees Celsius, but they grow even better like in the human body. So there's all these little kind of subcategories that we won't worry about too much. pH. Organisms typically will only grow in a fairly narrow pH range. Or we saw the temperature range can be quite large, but because that pH range, remember, is logarithmic hydrogen ion concentration, then again, usually a pretty narrow, maybe a point or two. There are organisms that grow best in really acidic conditions. So we call those organisms acidophiles. There are organisms that even grow near pH 0. There are bacteria that actually produce hydrogen sulfuric acid. There are organisms that can grow in our stomach where pH is about 2. They have ways to counteract that low pH. I think this picture is showing organisms that are growing in the runoff from a mine, acidic runoff from a mine. Most bacteria, though, most organisms in general are neutrophiles. They're going to grow best at neutral pH. Any disease-causing organism would be a neutrophile. And then, likewise, alkalophiles, or sometimes they're called alkalinophiles, grow best at a little higher basic pHs. An example there is the organism that causes cholera actually grows best at pH 9. So it's in nature, it obviously survives in our body as well. Okay, so the other thing that I mentioned is the oxygen concentration. And you may have heard terms like an anaerobe or an aerobe, organisms that have to have oxygen for their metabolism, or organisms that are poisoned by oxygen. So organisms that are poisoned by oxygen, though, it's actually not atmospheric oxygen. It's not O2 that is a problem for organisms. There's actually toxic forms of oxygen. Your book calls them reactive oxygen species, or ROS. And it's these toxic forms of oxygen that are damaging to cells, not just bacteria, but our cells as well. So I want to take a minute and think about, why are these forms of oxygen that I've listed there, why are they bad for cells? Where do they come from? And what do cells do about it so that they can live in the presence of those toxic oxygen forms? Okay, so first of all, why are they bad? They are all oxidizing or strong oxidizers. Let's put it that way. So when something's a strong oxidizing agent, that means that it's trying to fill up its outer shell of electrons. So it's grabbing electrons from other molecules, and that causes a lot of damage within cells. Can damage proteins, it can damage nucleic acid, pretty much everything. Okay, so that's why they're bad. Where do they come from? I'm going to highlight two that are the most common, but and by the way, don't worry about the chemistry here or the chemical formulas of these, but you should sort of recognize these as being harmful to cells. Singlet oxygen is produced during photosynthesis. So it's oxygen that's been kind of boosted up to another energy state. And organisms that photosynthesize have pigments in order to counteract that. It's the superoxide and the peroxide that are most commonly encountered, because those happen as a result of aerobic respiration. And we're going to talk about that more when that happens, when we get into metabolism, next lecture or the lecture after that. But during the electron transport systems, there's some kind of inappropriate splitting of oxygen. Sometimes we end up with superoxide as a result of the oxygen acting as the final electron acceptor. We might end up with superoxide instead of water. So that happens, we think, just about 2% of the time, just as a result of some, not everything working just perfect during respiration. So that we end up with superoxide and peroxides. So those are basically in the atmosphere as a result of aerobic respiration. Hydroxyl radicals those result from ionizing radiation. So cells are not going to commonly encounter that, so we won't worry about that. And the fifth form, hypochlorite ion, that's actually bleach, hypochlorite, and that is produced actually within our cells, our macrophages and neutrophils. That's one of the things that we can produce in our cells in order to kill the invaders that are phagocytized. So we'll mention that a little bit later on when we get to immunology. So a variety of ways that cells might encounter them, but they are in the air along with atmospheric oxygen. What can cells do about it? Cells have to have a way to detoxify superoxide and peroxides if they're going to live in the presence of oxygen. So they're gonna, how are they gonna counteract that? They're going to hopefully be able to produce enzymes, detoxifying enzymes. So superoxide, if I wrote out the equation here, which obviously I don't have memorized, superoxide dismutase is an enzyme that detoxifies superoxide into hydrogen peroxide and oxygen. Okay, so again, that's the enzyme. We'll write this over here. Produce enzymes to detoxify. Okay, so superoxide dismutase detoxifies superoxide into hydrogen peroxide. Unfortunately, this hydrogen peroxide, that's also toxic to cells. So they're also going to need another enzyme to detoxify that. One enzyme is catalase, and another possibility is peroxidase. Cells might have one or the other. Catalase is a reaction, again, that detoxifies hydrogen peroxide into water and oxygen. Okay, and that is a test that if you're in lab, you're going to do later in the semester. The way you do that test, you just rub a little bacteria on a dry microscope slide and then add a drop of hydrogen peroxide. If it bubbles, then the organism is able to make catalase. Okay, you look for a product of the reaction there. You're looking for those oxygen bubbles. Okay, so some organisms make catalase. Some organisms can make another detoxifying enzyme called peroxidase. Let's see. And that uses the electron carrier NADH. So you don't get oxygen from that reaction, so that doesn't test positive on a catalase test, but again, it's just a sort of an alternative enzyme. By that catalase test, we tell a big group of our gram-positives apart. The Staphylococcus make catalase and the Streptococcus don't. So that's a way that we can test to see which our cells might be. But so the important thing is if organisms can't make superoxide dismutase or catalase or peroxidase, they're not going to be able to survive in the presence of oxygen. So they would be some kind of an anaerobic organism. So there's a test that we can easily do. It's called the Thyoglycolate Test if we're trying to determine the oxygen requirements of a cell. And again, it's not really the O2, it's whether they can detoxify these toxic oxygen species. And you're going to do this if you're in lab next week. Again, it's called a thioglycolate test. There is thioglycolate in this medium. And basically what that does is it gets rid of the oxygen. Thioglycolate it actually reduces oxygen to water. So with any broth tube, there would be more oxygen at the top than it is at the bottom. But in these tubes, there's essentially no oxygen because we've chemically gotten rid of it. So we can inoculate the tubes and then look to see where the organism grows. If it only grows at the top, then it must need oxygen for its metabolism. And we could call it an obligate aerobe, or another way to say that is a strict aerobe. Okay, it can only live in the presence of oxygen. If it's only growing at the bottom of the tube, then it only must grow, only where there's no oxygen. Okay, an obligate anaerobe. There are organisms that are a lot of organisms are a lot more flexible than that. And the term facultative means flexible. So a facultative anaerobe is going to grow throughout the tube, probably a little bit better at the top. If it can do aerobic respiration and anaerobic respiration, it's always going to grow faster by some kind of aerobic respiration. It's always going to get more ATP when it respires aerobic. We'll talk about that in the next few lectures. Why don't we call it a facultative aerobe? I don't really know, but don't let that trip you up. They're always going to grow faster in the presence of oxygen, but again, they can do something like fermentation or anaerobic respiration. And then there's the aerotolerance anaerobe that doesn't really care if there's oxygen there or not, but it doesn't use it. Okay, so doesn't require oxygen or doesn't have any way to respire aerobically. It just does anaerobic respiration. It has detoxifying enzymes. It just, just grows the same throughout. And then there's this kind of small category of organisms that actually grow best with a little bit less than atmospheric oxygen concentration. So instead of growing right at the top, it kind of grows a little bit in a layer, just a little bit down. So again, grows best where there's less than atmospheric oxygen concentration. And we usually add an oxygen indicator in there as well, because a little bit, these have a cap on them, but a little oxygen does diffuse in. So you're going to assume that there's a little bit of an oxygen layer right at the top. Okay. Oh, and so we have ways to grow organisms in the lab. If they're anaerobic, it's definitely way more difficult. Some of our stock cultures, we just, some of them that grow a little bit better anaerobically, we might grow them in an anaerobe jar like this, where we include like a packet of chemicals in there that gets rid of the oxygen. Also, if you're working with strict anaerobes, you can work with them in what's called a glove box, where it's kind of hard to tell what that is from this picture, but you put your arms in it through there's gloves in there, and you can, inoculate your cultures or whatever in there. We have one of those upstairs, but it leaks for some reason. So it's never really been, I think somebody gave it to us. So again, it's just a lot more difficult working with organisms in that kind of environment. And as sort of a related note, the term capnophile is a term that you will hear if you're in lab later on. This describes organisms that grow best or maybe must have to grow a higher concentration than atmospheric of carbon dioxide. Okay, so they either grow better in high carbon dioxide, or they have to have elevated carbon dioxide higher than what's normally found in the atmosphere in order to grow. So we could, when we're incubating capnophiles, we do have a CO2 incubator. It's hooked up to a CO2 tank, and we pump a little bit more CO2 in there. Another less fancy way to grow them is with a candle jar. So guess a little bit more low tech. You put your plates into the jar, and it's kind of hard to see here, but this is a little votive candle. You light the candle, you put the lid on tight. When all the oxygen gets used up, the candle will go out. But the point is that combustion, that flame actually gives off carbon dioxide. So yes, it uses up the oxygen, but also gives off carbon dioxide, so we can get a little higher concentration of CO2 in there, and some organisms just grow better under those conditions, and we call them capnophiles. All right, a couple more things to mention here. The physical effects of water. That's why I reviewed osmosis when we were talking about membrane transport, because it's something that we'll be keep coming back to again and again. Osmotic pressure is something that's kind of difficult to define. You'll see a lot of different definitions if you look that up, but I like the definition: the tendency of a solution to take in pure solvent by osmosis. Okay, so if your cell is in some kind of solution, the tendency of the solution to take on water, okay, by osmosis. So that means basically what's the solute concentration of the solution. Okay, usually when we're talking about high osmotic pressure, we're talking about a hypertonic solution. So high osmotic pressure means hypertonic, high solute solution. Okay, so this picture is actually an aerial photograph of the Great Salt Lake. So that purplish color is due to the high concentration of bacteria, a certain kind of bacteria that's in the Great Salt Lake. There are organisms that actually grow best in hypertonic conditions, where there's some high solute concentration. And we talked about how that's usually bad for cells. They usually dehydrate, but certain ones have adaptations to survive that. The Dead Sea is another place where like 20% salinity. Normal ocean water is more like 3% salinity. So really, really salty places. The general name for an organism that survives these hypertonic conditions, we would call them osmophiles. So grows best in high osmotic pressure. And a halophile is an osmophile that specifically grows best in a high concentration of salt. All right, when you put something in brackets like that, that means concentration. So a high concentration of salt. Okay, so an organism that's a true halophile, it's actually going to be an archaea, probably not a bacteria. It won't grow like an isotonic solution. It needs that high salt solution. So that's very unusual. That's more of an extreme environment. Okay, so to wrap up this chapter, they don't talk about this in the book, but we have to think about ecological associations a little bit. If you're trying to grow a certain organism in the lab and you can't get it to grow, maybe it's because it's in pure culture and it needs to have other certain organisms around it. So it's a really hot area of study. We're trying to understand what kind of organisms are in our bodies, our gut, our large intestine especially. And it's very difficult to figure out what organisms should be there and which ones are actually making us sick, because there's so many of them. And again, they're in this community, so they're going to act differently. But we could use three broad terms to just describe relationships between organisms. Antagonistic relationships, that just means that one organism is harmed and the other benefits. Okay, so parasitism, that's an example. And you can think of lots of examples of that. Here's an example of a fungus, probably some contaminant that's growing on a plate of bacteria. You can see there's a zone around that fungus where the bacteria is not growing. That fungus is secreting something that's keeping the bacteria from growing. So one organism benefits, the other one's harmed. A synergistic relationship is both organisms benefit from their relationship, but they can live separately. And a symbiotic relationship, both organisms benefit and they do not live apart. Like the bacteria that lives in a cow's rumen that digests the grass for them, or in a termite guts, or the rhizobium that lives on the nodules of a pea plant that fixes nitrogen for the plant, and the pea gives it its nutrients. So, okay, we're out of time there. We're going to start the metabolism chapter on Wednesday. Have a good day.